Ladies and gentlemen, it is my pleasure to welcome you to Israel, ground zero for the cannabis industry. So let's have a round of applause for everyone coming in from all over the world. My name is Cheryl Shuman, and I'm from the United States. I'm the founder of the Beverly Hills Cannabis Club, and I am a media expert. And I'm very pleased to say that in the past 2 years, we have generated over $1 billion worth of earned media to rebrand cannabis for the righteous and a beautiful scientific plant that can heal not only a multitude of illnesses and heal the world, it can heal the economies from countries all over the world. So it is my pleasure to be here. I consider Israel to be part of my home. And one of the main reasons is I was a cancer survivor, and cannabis gave me my life back, and it was Israel that gave me the credibility and the integrity to go forth and spread that message around the world. I want to tell you a little bit about Saul Kay and this event. This is an event unlike any other in the world. It's the very first time in the world that we've been able to gather the top researchers, the top scientists, the top technology people, the top entrepreneurs, and the top investors from all over the world. We have people from over 40 countries here today that have come far and wide to be a part of Israel-cannabis and a part of CannaTech for 2016. But most importantly, I want to introduce a man by the name of Saul Kay. Saul Kay, who is the CEO of CannaTech and Israel-cannabis. This is a man, Saul, that I consider to be a part of my family. And you know, sometimes you meet people and you know the minute you meet them, you're going to be friends for life. And I am very proud and honored to be a part of this family. And this is the visionary, the man who put forth this event. So without further ado, I would love to introduce Saul Kay, who is the founder of this event.
G'day, good morning everyone. Welcome to CannaTech.
It's okay.
I started this cannabis journey 2 years ago with my partner Jason, who is a patient, and together we built a team. And we had one purpose, to showcase Israeli innovation in cannabis. What I found, I like to call C. Israelis have a lot of C, and we all think we know what I'm talking about. It has advanced us further and faster than everyone else in biomed, pharma, fintech, agtech, medicine, and even food. C is chutzpah. We had a researcher with enough chutzpah to ask to study a plant. We had patients and parents with enough chutzpah to demand a treatment for themselves. And we had a regulator who was willing to listen. And that is why we're here today in the only place fitting for this event, Israel. My dream was to showcase innovation and to start a global platform for collaboration, incubation of ideas, and a vehicle to invest and nurture this industry. An industry that has expanded faster than anything I've ever seen before. It's the first economy born in the digital age with an existing user base for its products. And it's a global economy. We are providing a platform that today and tomorrow will show you why Israel is key to innovators, researchers, and investors. So what makes it so special when you add Israel into the mix? Well, we've been at it longer, we do the research, we have an established record of medical discovery, and we made the desert flower. It's now my honor to introduce our keynote speaker, a man for over 50 years who's been a revolutionary in the cannabis space. Professor Mechoulam is acclaimed for his research. Yes, he discovered THC and continues to lead the way in endocannabinoid research. But for me, the one word that embodies more than anything Professor Mechoulam, and I've only met him a few times, is a mensch. Never in my life have I met an individual who is so humble, willing to share his knowledge, time, and expertise. A man who engages with open arms, with researchers, entrepreneurs, visionaries. And I've no doubt that many of you in this room have felt this and experienced this firsthand. Professor Mechoulam is a pioneer, a visionary, and an extraordinary human being. I'm humbled and grateful to introduce our keynote speaker, organic chemist and professor of medicinal chemistry at the Hebrew University of Jerusalem, Professor Raphael Mechoulam.
Thank you for inviting me. I hope that you'll enjoy your stay. I hope that we shall all be able to learn from each other. And let's discuss the various aspects of cannabinoid research, cannabinoid clinical applications, and cannabinoid use in society. And I'll try to present some of the topics that have been advanced in research over the last, I would say, 40 years. And now, I was supposed to have, and do I see it here? Where do I see it? I don't see, okay. When we started research on cannabis, there was absolutely no interest at all. Actually, when I asked for a grant, I was told, in the US, we asked for a grant from NIH, that it's not relevant to the US. Cannabis is not being used in the US, and when I have something more relevant, please contact us. Well, there is a little bit of a difference today when Nature, one of the major scientific journals today, asked me to write a review, and we did so. It was put on the cover, as you can see, as a brain and cannabis coming out of it, and they made a big fuss about it. Well, this is because it has become relevant, and the reason it has become relevant is maybe on the next slide. Some good friends of mine wrote a review on the medical aspects of cannabis, and they say that modulating endocannabinoid activity may have therapeutic potential in almost all diseases affecting humans. And this comes from two very distinguished researchers at NIH. Now, I haven't heard anything of this sort for any other neurotransmitter. Not for the major neurotransmitters like dopamine, serotonin, and so on. Nothing, to the best of my knowledge, there is no system for which one can say that it's involved in almost all diseases affecting humans. And therefore, whether it's correct, or 80% correct, or even 50% correct, it is of major importance, and this is why there is so much interest, scientific, medical, clinical, and commercial at the moment. So I'll try to give an overview of the scientific and clinical knowledge today, and I hope that we shall all learn from each other on the various aspects, including these that I shall be talking about. Well, let me start by going back a couple of thousand years ago. The Assyrians already used cannabis for essentially all those things that we use cannabis today. They used cannabis for, they called it ganzigunu, which is taking away the mind, which is probably a better definition than causing it high. They use it as a medicinal agent, and they use it in clinical, well, whatever they believe their gods were. Then if we move a couple of thousand years ahead, the Greeks and the Romans did not know that cannabis can be a psychoactive compound, a psychoactive material. They used it for inflammation alone. And nobody seems to have paid attention to that. Obviously, at that time, cannabis in Europe did not contain the psychoactivity, the psychoactive molecules that we know that exist today, and that's why they used it for therapeutic purposes alone. And if we move another few thousand years ahead, this is something written by the physician of Queen Victoria in the UK. She apparently suffered from migraines, and the British call it migraines, and he used to import cannabis from India, and she was Emperor of India, of course, because of her migraines. So we can see the various types of activity depending on where the material comes from, and obviously today we should know what the constituents are. Many years ago, about 50 years ago, we decided, I had just been appointed to, and that was at the time at the Weizmann Institute, then I moved to the Hebrew University. One of the topics I wanted to study were natural products, and one of the natural products was cannabis. And I was surprised, very much surprised, to find out by reading literature in English, in German, in French, in Russian. When one looked at the literature at that time, I was very surprised to find out that it was far behind the knowledge we had on just the opiates or cocaine and so on. Morphine had been isolated 150 years previously. But here, apparently, from a modern point of view at that time, the active compound had not been isolated. Now, this is not just a chemical game that the compound had not been isolated. If there is no precise definition of what the active compound or compounds are, it's impossible to do pharmacology. It's impossible to go into the clinic. Today we need exact knowledge. Medicine is quantitative. There is no such thing as take something and go away. When you go to take a drug, you have to know exactly what you're taking and under exact conditions. And the physicians will not let you just take something without good supervision. So as long as the chemistry was not known, it was impossible to go ahead. So at that time, we got cannabis from the police, and I always said thank you to the police in my publications. Not very seldom one sees a thank you to the police in scientific journals. We got in Saul's, the name Saul, in which the cannabis was smuggled, comes from the idea that the hashish was put into these kind of bags, put under soles in shoes, and smuggled that way. So we looked at these, at the chemistry, and I realized that many in the audience have forgotten the chemistry they knew. We found that there are quite a few compounds. They're more or less of the same type, and that was one of the reasons the material had never been, the active compounds had never been isolated in pure form, because it was a mixture and the technology was not there to separate the different compounds that were in the mixture. So we isolate a huge number of compounds, and those of you who can see the structures, they are more or less the same. But surprisingly, although there are quite a lot of compounds, and each one of them is a individual, if you wish, on its own, and can do all kinds of other things, or from a chemical point of view, of course, they are different. There are only two compounds, at least at that time, we realized that there are only two compounds that of this family of compounds which are of interest and which are present in large amounts. And this is tetrahydrocannabinol, the one of the two compounds that you see, and the other one is cannabidiol. And when we tested these two compounds, in great detail actually, we found that only one of us caused the psychoactivity and the other did not. But nevertheless, there's two compounds of major interest, each on its own. So we went ahead looking at the compounds. We tested the compounds at that time. A good colleague of ours at a biological research station in Arizona was testing it in monkeys, and the compound, the THC, caused the monkey, with one of them, it caused drowsiness in the monkey. The other compounds did nothing. So we went ahead mostly working with THC, and we and many other groups that joined us, not many, but a few groups that joined us all over the world. We looked at the chemistry and the pharmacology and at here and there and some clinical effects and metabolism, and we looked and we learned quite a lot of it. The one thing we didn't know, and nobody knew about it, how does THC act? It didn't act by the mechanisms that were known at that time. It was different. And a group in England said, well, it has a non-specific activity, and therefore for 20 years nobody really was terribly interested because it was non-specific. Well, we showed that it is specific by some other, I wouldn't go into the details, of course, and then a group in St. Louis, Alan Howlett, a good friend of ours, found that there is a receptor. And the receptor is found in all those areas of the brain that one can expect them to be. There are the names of the areas in the brain, but these are areas that have to do with movement control and all those effects that we know are associated with cannabis. So obviously it was part of a system. It was part of a very specific system which was unknown previously, and then we decided to look for the compounds that our body, our brain makes, because new systems, endogenous systems, don't exist because there is a plant out there. What happens is very often that there is an endogenous system, and this endogenous system can be mimicked by one plant or another plant, but it is an endogenous. It's regulated within our body, within the mammalian body, by compounds that we produce. So we went ahead looking for the active compounds, or for the compounds that stimulate or cause the effects of this through these receptors. At that time already we knew that there are 2 receptors, and after quite a lot of work we found that there are 2 compounds. The first one we called anandamide, the second is known as 2-AG, and these 2 compounds, you can see the structures, and you can see that these compounds differ completely, chemically differ completely from the plant, from the compound from THC in the brain, in the plants. And this was not surprising. We don't have to. The plant mimics the activity of the compounds, but not necessarily their structures. The only thing in common, chemically at least, was that both are lipid soluble. They don't dissolve in water. This is important because this is what happens in the body. They are lipid soluble compounds, and they bind to the receptors more or less to the same extent. They activate the receptors, both compounds, at more or less the same levels, with one major exception. While the plant compound stays there for a relatively long time, the compounds that we produce, the endogenous compounds, and anandamide and 2-AG, they are formed when needed. They are not present in the body all the time. At least one of them is not present in the body at all. Very low concentrations present there. And they are formed when needed, pain or something else. They are produced where needed. They act when needed. And they're immediately broken down by enzymes that are specific for them. So here we have a difference. Surprisingly, or not so surprisingly, anandamide has never been administered to a human because of all the regulations that exist and the money involved to introduce a new compound in the body. anandamide, a compound that we produce. And it's very important, and the same is true for the other one, 2-AG. They have never been given to a human. We don't know how they act in a human. That's a shame, but we can see that throughout research in cannabinoids. We have diseases that many people take cannabinoids for, but there are no clinical trials. We have the compounds that we produce and we know are not toxic, but they have never been tested in humans. So there is a lot to learn. But let me go ahead with the science part. So here we have a new system, new biological system, which involves the receptors, the compounds that we produce in the brain and in the body, different parts of the body. We have the enzymes, those that form these compounds, and those that break down these compounds, which are extremely important, because one can affect the concentrations of, let's say, anandamide, and by acting on the enzymes that produce them, or on the enzymes that break them down. And quite a lot of companies and quite a lot of academic institutions have worked on these compounds, and in this way they can play around with the levels of anandamide, which seems to be extremely important. I'll be saying a few words about schizophrenia. There, Cannabidiol, the second compound of importance, and there we know that Cannabidiol enhances the activity of anandamide by having more anandamide, by lowering the activity of the enzymes, and therefore we have a lot of anandamide in the brain, and it acts on schizophrenia. Again, very, very little clinical work has been done. Today we know that at least in some of in all these things that I mentioned here on this slide, and endogenous compounds and endocannabinoid system act, starting from anxiety, appetite, etc., blood pressure, pain, down to stress, and I can probably add another 10 or 15 additional situations. So that's one of the reasons that my good friends at NIH have written down that it is associated, affects almost all human diseases. I will not obviously go into the biochemistry, that's not something, I just want to mention one thing. Most neurotransmitters, compounds that are of extreme importance for what's happening in our body, and obviously what's happening during disease, the neurotransmitters are transmitted in the nerves from one part of the nerve to another part. The nerve is not a string that goes from the brain to the foot. Nerves are broken down at certain places, synapses, so that the body can regulate the activity of the nerves. Now, this regulation is caused by neurotransmitters, and they are placed on the incoming part of the nerve, and then when needed, they go to the second part of the nerve, the post-synapse. Now, these are extremely important physiological situations, and any major change there can cause a disease. If, under certain circumstances, dopamine, one such neurotransmitter, is not enough, it can cause one kind of disease, Parkinson's. If there is too much in a certain part of the brain, then what's schizophrenia? They have to be well regulated. Well, it seems that the cannabinoid system is a regulatory system in addition to everything else it does. It is formed on the second side of the post-trauma, the post-synapse. It goes from the post-synapse when needed to the pre-synapse. There it acts on the receptor, etc., and it regulates the other, or many of the other neurotransmitters. So this new system, the endocannabinoid system, is a regulatory system, and that's why it can act on so many things. Well, I'll just give a few examples as much as time allows me. Let's say neuroprotection. We found quite a few years ago that after brain trauma, one of the compounds, 2-AG, goes up nearly 10 times. Is that just because the brain trauma mixed up the brain, or the brain doesn't know what to do? Or is that a reaction of the brain to the trauma trying to lower the damage? Well, we synthesized the compound and gave it to mice that had undergone brain trauma, and yes, the amount of damage that was done, and the amount of damage is the white parts of the brain that you can see in the slide. When we gave them 2-AG, lots of 2-AG, it went down 50%. So here we have a compound, 2-AG, which helps in brain trauma. Unfortunately, like with most other things, it works excellent in mice, and it has never been tried in humans. I hope that someday somebody will have the material and the opportunity to do that. It's a pity, because there is nothing really, again, no drug, a very effective drug in brain trauma. Now, there are two receptors. One of them is Cb1, the one that causes the psychoactivity, and there are quite a few things that enhancement of this this part of the endocannabinoid system, this receptor. For example, we all know that it prevents the side effects of cancer chemotherapy. We did that in the clinic, actually, with Professor Avramov about, I would say, 20 years ago, and it was extremely effective in children, extremely effective. We knew we could block essentially all the side effects of cancer chemotherapy in children, and we didn't see any side effects. It also enhances appetite in feeding. Normally, this is important in, let's say, AIDS patients. We recently published, and this was the work of the psychiatry department at Hadassah, and Dr. Reutman was in charge of that. He found that in a small number of patients, it was possible to lower the effects of post-trauma. This has been known. I mean, people have talked about it, that there are no clinical trials. He showed it in a clinical trial, well-done clinical trial, a small number of patients, that most definitely it helps with the post-trauma, especially with sleep. Many of the post-trauma patients cannot sleep, and here he saw that it affects many of the side effects, not the side effects, the basic things of post-trauma, including sleep. What about the Cb2? Cb2 is more interesting. The Cb2 receptor, the activation of the Cb2 receptor does not cause any of the high that we know about, but it seems to be a general protective agent, like the immune system. It's a second type of protective system. It works together with the immune system, but it's a second type. And it seems to be a protective system of a very general nature, and I can give 10 or 20 types of examples of activation of the Cb2 receptors. And now there are companies and some of compounds that we have synthesized which act particularly on the Cb2, not on the Cb1, and they promise to be extremely important new drugs because they're specific for the Cb2. What does the Cb2? It attenuates inflammation. It has been shown to act on all kinds of injuries, and wouldn't go into details, that we know about. And other companies are working with Cb2 specific compounds on eye problems, specific compounds on liver problems, specific compounds on other. So I expect that within the next 10 years, maybe less, we shall have cannabinoids on the market which act specifically on the Cb2 receptor. They will not cause any anything resembling the high. They will be specific protective agents for certain diseases. One sees that in Crohn's disease, one sees that inflammatory bowel disease, and we shall see here Dr. Timna Naftali speaking about these diseases, and it has been shown in asthma. What about cannabidiol? That's even more interesting because cannabidiol does not cause any of the central effect that THC causes. Well, we know that it acts on anxiety. There is even a small clinical trial. I mentioned schizophrenia. One needs extremely high doses, about 1 gram a day, but it acts, and maybe, just maybe, it will be used in schizophrenia. It causes no side effects. A group in Germany has done a clinical trial in schizophrenia, and the effects are outstanding because it causes no side effects, while the anti-schizophrenic drugs on the market all have side effects. We have seen in animals that it works in diabetes type 1. We have a strain of mice that develop diabetes type 1, and all of them develop, but if you give them the cannabidiol, only 30% develop. And there are lots of numbers, but if you look at the numbers on the percent of damaged cells, those animals that did get cannabidiol, only 7% of the cells were damaged, the relevant cells that produce the insulin, while those animals that did not receive the cannabidiol, you had 77% that had those damaged cells. So here you have something very specific, very important, and probably the same as in humans. What happens in humans? I have no idea. Nobody has done the clinical trial that is obvious here, because it works so well in diabetes type 1, and there are many patients, children, that have diabetes type 1. I wish a clinical trial should be undertaken, but obviously a group like mine cannot do it. Just a couple of months ago, Dr. Yeshurun at Beilinson published, we were slightly involved in that, published a paper on Graft-versus-Host Disease. You may be aware that during, certain cancer diseases, the bone marrow has to be replaced. Well, the body doesn't like to have its bone marrow replaced, and the bone marrow that's replacing replacing the bone marrow doesn't like the body, and so they fight to each other, and the patients, many of them are very sick. Well, it turned out that if we give cannabidiol, which is I wouldn't go into the details why Horowitz did that, Yeshurun did that, it helped in about 50% of the patients. There are very few drugs of this type available. And I strongly believe that it will be of great help. And Dr. Yeshurun is continuing his work, trying now on more patients. He tried it on 50 patients, and I hope that now he will get even more results. So what about epilepsy? Well, we have an interesting story here. We tried epilepsy in animals, and then we tried epilepsy in humans. We were able to do a clinical trial, a small one, 15 patients. We did that in South America, and it was an extremely potent compound without any side effect. The doses needed were high, and we gave it to patients that nothing else was helping anymore, not to children. We couldn't do that in children, obviously. And we found that indeed it works. We went back to the old literature, and yes, it's there in the literature. It has all kinds of, there are all kinds of examples. Here is an example that was discovered by the person I knew, Professor Rosenthal, who looked at some Arab manuscripts he found in Paris, and he found that the epileptic son of the Chamberlain of the Caliphatic Council in Baghdad had epilepsy, not too many years ago, 1464, and he was given cannabis, and he didn't have any attacks anymore, but he had to take the cannabis throughout his life. Well, our results were more or less the same. We got very good results. And in 8 patients that were given, and there were 8 controls, 8 patients that were given cannabis, 4 had no attacks, 3 had very few attacks, 1 was not affected. So this was a clinical trial that should have been repeated 35 years ago, was done by us. It was not until the last 5, 6 years, parents discovered that children that get cannabis or cannabidiol can be helped. So they started going to Colorado where they could get cannabis, and now they are treating their children with cannabis that helps them, that helps many of them. Now, why did we have to wait 35 years, and yet there is no published major trial? There are several major trials going on in the US because of the public pressure that, well, here we have results, why isn't the government doing anything about it? So this is what's going on in the States, and it seems that, yes, they are reproducing our results of 35 years ago. We didn't have to wait 35 years ago. Too many patients have suffered. So what about cancer? Well, there isn't a single cancer clinical trial that has been published. There are thousands of people that use for various types of cancer. It seems to be helping some of them. Maybe it helps in all of them. Maybe it helps only in some cancers. We have no idea. We only have individual reports by patients that have taken, I'm not sure what they have taken. It seems to be helping. This is not the way to go ahead with in the clinic. Maybe we need high doses of cannabidiol for certain diseases. It seems that prostate cancer is an important goal. It seems to work there. I'm not sure about others. I'm not sure whether it works so well in glioma. Maybe cannabidiol, looking at what's in the literature, maybe cannabidiol helps some of the drugs that are being used in glioma. Maybe. But we don't have the data. Maybe it works in animals, but we are not mice. So it should be done, but clinical trials are missing. So one way that the drug industry goes ahead, it takes a well-known drug or well-known natural drug that has its drawbacks, the doses are high, and so on, and makes derivatives. Penicillin was an excellent drug when people started using it. You cannot buy penicillin anymore. You can buy a derivative of penicillin. The same is true for cortisone. You cannot buy cortisone. You buy a derivative. So chances are that we shall have new cannabidiol-type molecules. And I want to show 2 examples, 2 molecules, types of molecules. We made a lot of them, which are being developed by companies. This is one of the compounds, a cannabidiol with a fluorine, and this is being developed by a company called Phytex, and I hope they will be successful. And there are a few other compounds of this type. And there is another company that is developing an anandamide-type compound. Wouldn't go into the details how and why we discovered that this type of compounds are extremely good, some of them in osteoporosis. An anandamide compound, oleal serine, a fatty acid bound to a derivative of an amino acid. So we took this compound, which we discovered and which we published a few years ago, showing that it's an excellent anti-osteoporotic compound. We took this compound and made some chemical changes, so it becomes a better compound, more stable compound. And this compound is now being developed by another company. I hope that it will be successful. We shall be hearing about it, about some of the recent developments by Yossi Tamp, a colleague of mine. And I sincerely hope that we shall be, it will be new compound that helps osteoporosis and osteoporotic type compounds. Well, in summary, the endocannabinoid system is of extreme importance scientifically, medically, clinically. I hope, sincerely hope, that there are going to be clinical trials. We have to work on better chemistry, better clinical trials, and we shall be looking at medicine, cannabinoid medicine, the way we look at standardized medicine in other fields. And there should be a separation between the medical aspects and the social aspect. These are two different things. They should be looked from a different point of view. And I want to thank my colleagues who have collaborated in Jerusalem and everywhere else, and many of my collaborators in many countries. And I want to thank you.